We're very, very happy to welcome you. Today, we're going to have a fantastic session with our speakers, Austin Burt, Tony Nolan, and Abdoulaye Diabate. I am hopeful that we have Dr. Willie Tonui on the line. I'm trying to find his name on the list here, but I can't. He is going to be joining us as well from a regulatory perspective. Willie, if you are there, kindly turn on your video as well, because I can't find your name on the chat line. In addition to our main speakers, we have an army of volunteers on the line. And these volunteers are experts who have worked on gene drives as well. And they are going to be supporting with Q&A, but also, triaging some of the questions and bringing them up for our main speakers to handle. And I would like to say we're really, really grateful for this army of experts. We have Kairos, we have Tony, can you remind me the names of the experts from Liverpool?
We've got Charlotte Quinn.
We have Charlotte
from here at Liverpool School of Tropical Medicine, and Kiros Kiru from Imperial is also helping.
Brilliant. And then we have Delphine Tizi, and we have my colleague Lina Finda is on the line as well. We are hoping that there are several other people who work on gene drives. And please, if you're on the line, kindly participate in the chat discussion as well. So as you can see, this masterclass is kind of oversubscribed, and as a result, we are also streaming live on YouTube. So it will be on our YouTube channel as well. Just for your information, we will be starting in approximately 2-3 minutes. In the meantime, if you guys have any questions, please ask. This is to the main speakers. If you have any questions, please ask. And Tony, if you don't mind, may we invite the 2 additional experts from Liverpool just to introduce themselves on video and then mute back?
Okay, I can see Kiros Kiru from Imperial. I can see him there.
Yes. So Kiros, if you're online, can you just unmute your video, just so we can introduce you. Delphine as well, Lina, and my other colleagues who are going to be supporting here as well.
Hi, here I have my video on, and hi, I'm Kiros Kiru, and I work at the Chrysanthemum Lab at Imperial College, and I'm here to help as I can today answer questions to my knowledge.
Thank you very much, Kiros. Do you have Charlotte?
Yeah, and Charlotte is here. She is a freshly minted PhD student, so she won't call herself an expert, but she's doing very well, and she's got a lot to offer and will do very well at answering questions.
Excellent. Excellent. And do you have Delphine online already?
Yes. Hi, Fredros. Hi, everyone.
Yeah.
So I'm Delphine Tizi. I was for a bit the stakeholder engagement manager for target malaria. And so I'm more an expert on engagement issue, but also policy issues. So I'll leave Kiros and Charlotte answer the more molecular, and I'll try to take the other ones.
Cool. And of course, if we have other questions that you really want the experts to deal with, we will do that as well going forward. The last thing I want to mention is please use the chat box as much as possible. Send your questions through the chat box. Just type them in. You notice that this is not a webinar, it's really a meeting. So there's a chance for deep interactions between the participants, and that is something that we really cherish. Welcome again to Ifakara Health Institute. Today, my regular co-host, Dr. Sheila Ogoma, unfortunately is unable to join with us. She will probably join with us in a few minutes from now, in maybe an hour from now. Right now, she's not able to start with us as usual. Otherwise, thank you very much. We will be starting in 1 minute. And as I said earlier, this masterclass is being streamed live on YouTube today as well. So for those people who might want to, observe this or share with their colleagues, They are welcome. I would also like to take this opportunity to announce that next week we will have our 19th session of the masterclass, and it will be delivered by Professor Hilary Ranson from Liverpool School of Tropical Medicine and Professor Corinne Gofford from the London School of Hygiene and Tropical Medicine. So we will have a fantastic masterclass next week on how to kill mosquitoes. Really, the theme will be the mosquitoes must die. It will be focused on new interventions that can be used to counter insecticide resistance. So again, you're all welcome to that masterclass coming next week. All right, so at this point, I'm going to request all our other participants to kindly mute your videos except for the main speakers. And again, to make a call for Dr. Willie Tonui, if you're online, kindly unmute your video and be with us. We will be starting in 20 seconds. Austin, how are you feeling?
All right, yes.
Good morning.
Looking forward to it.
Ladies and gentlemen, my name is Fredros Okumu. I'm from Ifakara Health Institute, and welcome. We're going to begin now. And today's masterclass, as you can see, we are asking a very, very difficult question, but also I am told a very simple question. So if you don't mind, Tony, I would like us to begin this conversation from your perspective. I think the biggest question that my colleagues asked me today to ask you is this idea of whether we should worry about the gene in our mosquitoes. So there is a gene in our mosquitoes, Tony. I know it. I think you know it. I think everybody knows it. Should we worry about
it? Should we worry about it? Well, let me answer that another way. I initially understood this to be a question. Is there a gene in my mosquitoes? Mm-hmm. And the short answer is yes, there are thousands of them because, you know, mosquitoes, just like us, are made up of cells, and in those cells there is the genetic material, which is the DNA, and that DNA is organized into units called genes, which are responsible for the production of traits, so things that make the organism appear or behave the way it does. So in a way, it's a sort of a provocative question, but I think it's actually a very useful one, because it goes to the point that not everyone's understanding of the basics of genetics, or even the principles of DNA encoding things, is not clear for everyone. And that depends on the general literacy in this type of techniques. And it is a challenge to try and explain something like gene drive, which probably we'll do in a minute, when you don't know what the underlying level of understanding of the principles of cells, DNA, chromosomes, genetic units, etc. So it's challenging.
Tony, typically when people speak about gene drives, they think of something artificial. They think of something that someone has put in the mosquito there. Is it accurate to say that we also have natural gene drives that exist?
It is accurate, yes. So, you know, DNA encodes the genetic material, and those genes that are encoded within that genetic material are transmitted to offspring. Genes that get transmitted to more offspring do better and propagate an increase in the population. And you can get selfish types of genes which propagate themselves, sometimes at a cost to the organism, but as long as they increase in frequency each generation, they do better, and they're highly present in the population. And there are natural examples of that. And those are, you know, in some cases, natural examples of gene drives. So genetic elements that increase in frequency each generation.
Thank you very much. We're going to have a very long discussion on this subject. So before we dive deep into the subject of gene drives itself, I have a question for my colleague Diabate. Abdullah Diabate, if you're online,
Yes, I'm here.
Here is a question for you that has come up a lot in our recent masterclasses here. It's also a very general question, but an important one. This is with regard to malaria control efforts in Africa. A general question for you as someone who works in perhaps one of the most difficult countries in terms of malaria control, most malarious countries. What are we doing wrong? Why are we not at zero yet?
Well, I think this is a really tough question for us. Because if you look across Africa, basically, you have some countries where the statistics really are maybe on the side. And unfortunately, in some other places. So...
Yeah, but your sound is not very clear, if you don't know.
Oh, yeah. Okay. Can you hear me now?
That's fantastic.
Yeah, okay. So I was saying that if you look at the statistics across the landscape of Africa, so you will realize that in some countries, basically, I mean, the statistics are really on the downside. But unfortunately, if you take some other country, like Burkina Faso, for example, you will realize that the statistics really, unfortunately, the malaria case is really, getting higher every single year. So the reason for that, I think, is really very complicated. There is no single straight answer or thing that you can point to say that this is the reason why, we're not getting to the zero level. But from my experience, what I can say, if you look at the situation, for example, in Burkina Faso, and I believe also this is the case in most of the, 31 countries that are representing more than, the 95%. Hello?
Yeah, just speak up a little bit, Diabate.
Yeah, okay.
Yeah.
So I think you have several reasons, but the first obvious one for me, even though we don't have a specific empirical data really to show clearly that this is the reason. First of all, I think the problem is that you have a lot of insecticide resistance, because if you look at the current tool, we don't have that many tools, unfortunately. So most of our tools basically are using chemicals. Do you do IRS? It's with chemical. Do you do Bennett? It's with chemical. Even larval sort management, sometimes also you have a chemical that you use. So this means that, we have all our strategies that are based on only one element or tool that we are using in the field. And unfortunately, we're going to fire back, because if you look at the time when malaria, the implication of mosquitoes in malaria transmission has been detected to up to date. So we have really very few tools that we are making sure that we can use. But we know that you have that much plasticity in mosquito genome in a way that if you come with a strategy, mosquitoes always have time, to adapt. So if you take the case of Burkina Faso, for example, I can tell you when I was doing my PhD, at that time, insecticide treated bed net were just coming up. But if you look at that time at the resistance profile of insecticide resistance in Burkina Faso, it was really very low. Very few pockets in Burkina Faso that you could see the resistance. And not only that, but even the level of the resistance in these villages also was really, very low. Come back today. There is not a single place here in Burkina Faso where you can go and then collect mosquitoes, expose them to this conventional insecticide that we have adequately, and you will not find a resistance. So you have resistance pretty much everywhere. How much is it impacting now, the effect of malaria transmission is not really very clear, but for something sure, I think that it's impacting at some level. But the second element also, I think is really very important, is the health coverage. If you take many places in West Africa here, if someone is sick, so you will have, to drive for maybe 10 kilometers before you get to the first health care. And so if the healthcare is not really at the disposal of people so that they can quickly get access to it, so it's become really very difficult. And then also remember that malaria is something which is happening during the rainy season, where most of our people are really in the far. And so the breaking, and then so if you are sick and then you don't have even a way, to take your kid to a healthcare, then it's become really very difficult. And then the cost also is associated with it, even though most of the things that are subsidized now. But still, I think all these elements are part of it. And then the last element also, you will be really very surprised as of today, unfortunately, that if you go to some places in Africa, you ask that, do you know what is the cause of malaria? you will have various answers that will make you think really that even today, so people do not have a clear understanding of where is the cause of malaria. So I think all these factors are playing in, and this is one of the reasons why it's extremely difficult. But to finish, now I know that I'm taking
you got to finish it.
To finish on that, I think that it's extremely important that we critically look at these different components. But if we don't have any tools, we won't be able to find. So given that the tools that we have in hand today are really very limited, so there is a critical necessity, to come up with new tools. And this is exactly what we are doing in Target Malaria.
We're going to be talking a lot about that. I want to go to Austin now.
Ross, one second.
Tony.
Yes,
there's
please.
a problem with Diabate's audio. It's difficult to hear.
Oh,
yeah,
so it would be better to sort that out now. Otherwise, we'll lose the interest of people.
Okay. Can you hear me? Can you hear me now?
We can hear you now, but please take some time to sort out your video, and then we will be with you.
No, the video is fine. It's the audio.
Your audio, I mean.
Okay. I think that I'm in a confined small room like a prison. There is an echo, probably.
So please sort it out, and then we can proceed.
Okay.
We can hear you, but that's fine. Just take some time, and then we'll be back with you.
Okay.
Take some AirPods or something.
Yes. The other thing is we are requesting everyone else to kindly mute your microphones and mute your videos as well. While we're doing that, Austin, two weeks ago we had a masterclass with Professor Brian Greenwood, and we asked him in the end, what he really thinks are the two tools that have greatest potential. This is what Brian told us. He said that in addition to improving resilience, in addition to kindly mute your microphones, please. Everybody there, otherwise I will remove you from the call. Excuse me, guys, just one moment. Tony, are you able to unmute?
Yep.
Oh, brilliant. So I've just muted Diabate, and we'll figure it. So Austin, Brian told us that if you leave aside everything else that we know, according to him, the two things that have greatest potential are gene drives and vaccines. And of course, vaccines are, we have a fairly good candidate, about 30–40% efficacy being trialed right now, RTSS. On the gene drive side, we have a product that is only proven in the lab, not yet proven in the field. The question we have for you is, do you share the perspective that Brian has, that gene drive fits that bill as a potentially revolutionary tool that would really drive us towards zero beyond everything else that we have?
Yes, absolutely. I unfortunately missed that masterclass with Brian, but from what you've described, absolutely. I think gene drive could be quite a transformative tool in terms of getting on top of malaria and driving it, as you say, to zero. It's quite a different, so as you know, and as I see from the title on the slide here, halfway there to elimination. So the existing tools of bed nets and IRS, the chemical tools and the drugs have been very successful, saving millions of lives. So that's great, but this consensus seems to be that the existing tools are not enough to get us all the way to zero, that something transformative is needed. Yep. And yes, gene drive or a vaccine, and it would have to be quite an efficient vaccine, could do that. So yes, we're quite excited about the potential for gene drive to really make a transformative contribution. Won't do it all by itself, but a transformative contribution to getting to zero.
You are a global expert on this subject. You're probably one of the people who works on this longest. And for that reason, I would like you to actually help us with the basics here. So just for everyone, for people, my colleagues on the line, people asking really, where does this all fit in? I would like you to help us with the basics in the sense of what exactly is gene drives and how does it compare to all the other biological control tools? How does it compare to sterile insect technique, for example? I've noticed in one of your writings, you describe this in a group of the so-called heritable strategies. And it would be nice if you could start from there and just explain to our audience what this really is.
Sure. Okay. So within the heritable or genetic or biological approaches to malaria control, or indeed any kind of pest control. So the first approach that has been used now for decades for semi-agricultural pests and is the release of sterile males. And for example, I think it's also been used for tsetse flies in Zanzibar, unless I'm mistaken. So that's to, you basically inundate a population with sterile males. They mate with the females of that population. Those females don't produce any progeny, and so you suppress the population. And so, as I say, that's been used for some agricultural pests quite successfully. And that relies on inundation. So you have to essentially swamp the population with 10 times or 100 times more sterile males than there are natural males. And in that way, you have to do that repeatedly. For many generations, and in that way you can suppress the population. That approach works really, can work well when you can rear lots and when the population is relatively targeted, yeah. And you can rear enough in order to swamp the population. Gene driver, and so for example, that's the Oxitec approach to controlling Aedes mosquitoes, which is largely an urban issue, is to have effectively genetically modified mosquitoes that are effectively sterile, and again, to inundate the population and suppress them in that way. Right. That approach is unlikely to be able to work for malaria control in Africa because that's largely a rural disease, and there's just too many mosquitoes and too much space to hope to be able to inundate, to swamp by a factor of 10 or 100 the natural population. So we need, if we're going to use a genetic approach, we're going to use something different. And that's kind of the attraction of using a gene drive approach, is that you're not having to inundate the population. You just have to inoculate with a relatively small fraction, release 1% or 0.1% of the population to a release. And then over successive generations, the gene drive construct will spread and increase in frequency, 1% to 2% or 4%, etc., spread through the population. And then as it does so, well, there's two broad strategies people talk about in terms of how you might use gene drive for malaria control. One is to suppress the numbers of mosquitoes and so reduce transmission that way. Or alternatively, to change them, leave the numbers more or less intact and change them such that they're no longer able to transmit the pathogen.
So in your writings, the text that we put up on the slide is actually right from your documentation, from your, I believe this is your 2014 paper. Yep. The items number 3 and 4, this is what you refer to as population suppression being item number 3 and population modification being item number 4, right?
Yes. Number 2, a driving Y chromosome is another way to get population suppression.
Right. We will talk about that in a little more detail. But before we do that, I would like to jump to Tony, just to carry on with this explanation of the 2 types of gene drives that Austin was talking about. So Tony, in the beginning of the year, you published this fantastic paper in the Royal Transactions, Philosophical Transactions, I should say. And I've read many gene drive papers, but you remember I called you to say this is the first time I saw simple writing on this subject.
Thank you.
And I'm very grateful actually that you put this together. The diagram on the left, this is right from your publication. And if you don't mind, I see you have the Cas protein onto it, so it can also link really nicely to the description of CRISPR. Could you please take this from here, from where Austin left it, and explain how gene drive fits in and how you do these two approaches using that diagram?
Okay, yes. So I don't know if, can you see my cursor?
No,
not,
but
I'm not sharing the screen.
Yeah.
Is there a way to do that?
I can help you.
You can do. Okay, so if you see in the top left here, this panel on the top left, these two vertical lines represent mosquito chromosomes. So just like you and I, mosquitoes have 2 copies of every chromosome. Okay, and when they make offspring, they donate one of those chromosomes to the offspring, and the other parent donates one of their chromosomes to the offspring. Okay, so if you see this chromosome on the left here, this has a gene that we want to get into a population here, yeah? So this gene might encode some genetic trait that we want to spread into that population.
That's the gray one.
Exactly. So ordinarily, half of the offspring would receive this chromosome on the left with the trait that we're interested in, and half of the offspring would inherit the chromosome on the right, which doesn't have the thing that we want. Okay, so that means that every generation, only half the offspring get the gene and the associated genetic trait that we want. And so the way that a gene drive is different is that it alters this chromosome on the right so that it now also contains a copy of the gene that we want. So no matter which chromosome is inherited by the offspring, they all have that gene and all have the trait. So all the offspring get that gene drive and the associated genetic trait. And in turn, when those ones in the next generation, they mate, all of their offspring will get it. So gradually and gradually, all the offspring in the population start to get that genetic trait. Now, you asked me about the Cas9, this protein up here. That's just one way of ensuring that the gene drive is able to copy itself from one chromosome to the other. In this specific example, it cuts the chromosome, and when that chromosome gets repaired, it's tricked into copying the gene drive onto that chromosome. But there are other ways of ensuring we can get this biased inheritance. But that's the basic principle of a gene drive.
Okay, so what you're saying is you have the Cas9 protein, which is like a pair of scissors, right?
Yeah.
And this is the guide RNA, so just like to go into the chromosome and search for this specific part that you want.
Yeah.
And this is the part, the thing that you want to insert in there, the gray area.
Yeah, so sometimes the analogy that I use sometimes is think of a gene drive as a car that's designed to spread and drive through a population. In this case, the Cas9 is the motor that's driving that car. There are different motors that one can have, but in this case, it's Cas9, and that motor works by cutting the chromosome in a very specific region and ensuring that the whole element gets copied across onto that chromosome.
Brilliant. Thanks a lot, Tony. Diabate, we'll come back to you, but first, before that, I would like to request Austin to finish up on this discussion using a description that he and Andrea Cristanti put up a few years ago. So, I mean, Austin, this year, there was a nice publication by Luke Alfie, and I don't know if you are part of this group as well, in an attempt to define the concept of gene drives. However, reading back from some of your work, I see this first sentence from this paper that you published with Andrea, with Dr. Crisanti. And it says what I believe is perhaps a clearer definition of gene drive. So if you don't mind, can you talk about this preferentiality, this idea that at the end of the process, everybody has the gene that you want them to have?
Sure. So as Tony mentioned, mosquitoes like us have 2 copies of every gene, and normally one or the other is passed on, and it's 50-50 which one you pass on when you have children, and similarly for the mosquitoes. And the whole notion of a gene drive is that it's no longer 50-50. Instead, it's biased or preferential, so that so that it's passing it on to 70% or 95% or 99% of the progeny instead of the 50%. And because of that preferential or biased inheritance is what allows the gene to spread through the population, and indeed can spread even if it's causing some harm to the organism.
Right.
So most genes, of course, that are inherited in a 50-50 way can only spread through a population if they do something good for the organism, if they increase the mosquito's survival or reproduction. And so insecticide resistance spreads because of that. But this using gene drive is in principle a way that you could spread a gene through a population, even if it's harmful to the mosquito. And so that's what makes this process attractive as a pest control measure, is this ability to spread things that are harmful to the organism. And you can even do something that might seem counterintuitive and spread sterility through a population. If you ensure that females that inherit the gene from one parent are fine, but if they inherit it from both parents, then they're sterile, then that sterility can become more and more prevalent in the population due to a gene drive.
So you're saying that in standard fashion Mendelian evolution, if something is too costly, it will eventually disappear from the population.
Absolutely. So we only expect things to spread if they do something good for the mosquito.
Okay.
That's natural selection, that's the textbook natural selection, of which insecticide being resistant to chemicals on bed nets is one example.
So in the days of Darwin, this thing would survive only if it was helpful
to the mosquito.
To the mosquito.
To the survival or reproduction of the mosquito, yes.
All right. But you're saying that with gene drive, it doesn't matter. Even if it was harmful to the mosquito, it will still spread.
That's correct. Because of this biased inheritance, because it's not being transmitted 50-50, because it's instead getting into 95 or 99% of the progeny.
Yeah, forgive me for stressing this part, but a lot of colleagues on the line are not necessarily molecular biologists, and this is an important subject for especially my colleagues in Africa. We stand a chance to gain the most from this technology if it works. And it would be nice, if you don't mind, we're asking you guys several times to simplify stuff.
That's fine, it's very important.
Austin, on this paper, you also introduced these 3 terminologies, the chromosomal shredding, which I think you mentioned earlier about the Y shredding part, but also this so-called the homing endonuclease genes. And you have, I mean, this appears a lot, it's like the core of your expertise. You have described these HEGs, or the HEGs, as selfish genes. I think you guys in your labs, you use the term selfish a lot, but you refer to this as very selfish genes. Explain this.
Sure. So these homing endonuclease, so what Tony was describing before of having an enzyme, a gene that encodes a CRISPR enzyme that makes a cut on a chromosome, and then the gene gets copied across into the chromosome where previously it was absent. That's something that occurs naturally. There are naturally occurring genes called homing endonuclease genes that exist in many microbes. So like in fungus, yeasts, algae, other small organisms. And that's exactly what they do. It's just a gene that makes an enzyme that cuts a chromosome where the gene is not present and then gets copied across during the repair process. So, these were first described, they've been described in decades ago, first in yeast. And they don't do anything good for the organism, for the yeast or for the fungus, for the algae. They don't, in terms of increasing its survival or reproduction. They just are little tiny parasites, if you want, on the organism, just a single gene parasite, or also known as a selfish genetic element. Okay. So as I say, these things naturally occur. In a sense, what we're doing is trying to mimic this natural process to make versions of these sorts of, to use the same sort of process and adapt it to mosquitoes to try and control them.
Thank you very much for that. Now, two things now. You have the homing endonuclease, and then you have the Cas, and then I start to get confused. So I'm going to ask Tony to help clarify this, and I see Tony has a point to make, which you're welcome to do right now.
If you don't mind, Fredros. So I think this analogy of a sort of genomic parasite that's very specific for the mosquito in this case, or the whichever insect you design it against, is quite useful, because then I think it's easier to understand this concept that that genetic element is acting like a parasite in the genome of that particular species that you're trying to control, and then it's a bit easier to understand, perhaps.
Okay, thanks. I pulled up an image here from Kevin Asbell's publication, and put this aside, side by side, Tony's work. And the reason is, and forgive me if I misunderstand this subject, a egg is selfish. If you have the other allele doesn't have it, it cuts and it inserts itself there.
Yep.
When you then say Cas, so Cas and egg, what's what here?
Okay.
Explain these two things.
So I will do my best to explain that.
Yeah.
And actually, it's well framed, because when people hear of Cas9 and CRISPR in the context of a gene drive, they can sometimes make the mistake of equating all CRISPR-Cas9 applications with gene drives and vice versa. And that is a point of confusion. So if we look on the right here, apologies for biasing it towards my own diagrams.
That's fine.
So what leads to the copying of this element here on the left is the fact that it expresses Cas9 and the guide RNA that cuts this chromosome here, or the black spot on this chromosome there.
Correct. Mm-hmm.
So then we have a broken chromosome. Okay. Mm-hmm. So the broken chromosome at a very specific location is exactly what those homing endonuclease genes do in principle. They cut the chromosome at a very precise location. And so we can re-engineer the CRISPR-Cas9 to do exactly the same thing, to cut the chromosome in a very precise genomic location. And the way that this chromosome gets repaired, that leads to the copying of the gene drive element from one chromosome to the other, is nothing actually to do with this Cas9 element or the homing endonuclease gene. It's the way that the cell repairs the broken chromosome. And it does that by using this intact one as a template and copying all the intervening sequence. And so that's why the homing endonuclease genes, let's say, is the natural example of this. And the CRISPR-Cas9 version is, if you like, a sort of synthetic version that tries to copy that process and does work well. Okay, but the big advantage of this is that it's very easy to re-engineer CRISPR-Cas9 to recognize a particular site in the mosquito chromosome that you want to target.
So
is that clearer?
Means that's well, you have to confirm to me if we got it right. But so even if CRISPR-Cas hadn't been discovered,
yes,
or the eggs would still do the same thing.
Very good. Yes. So
we didn't
start working on. We started working on this before CRISPR was discovered, or at least known to be a reprogrammable DNA cutting enzyme. We were making progress on this, but CRISPR just speeds up this this throughput because it's much easier to rearrange in order to recognize specific target sites.
Makes a lot of sense. Thank you so much. Thank you so much. Let's keep this molecular biology business side for now. Diabate, let's talk a little bit. Something closer to home. You and me are vector biologists. And when I first heard about this, I had the same question that I still have today that many of my colleagues have as well. In the earlier talk, you were talking about putting this together with other interventions, putting gene drives together with other interventions. Now, gene drive technologies involve releasing mosquitoes. Our current core interventions involve killing mosquitoes. How do you put a technology that involves releasing mosquitoes together with an intervention that involves killing mosquitoes? Where do these complement each other? Is there a chance that IRS, for example, will kill all your mosquitoes as soon as you release them? We can't hear you, Diabate. You're muted. Unmute.
Yeah, well, okay, I was saying that that's a really, very good question. But in the first place, I think the most important thing, to mention is that we're coming up, with gene drive to try to control mosquito population in one way or another, malaria transmission, because the conventional tools that we have in place, there are not enough, to get rid of at least a high proportion of mosquitoes so that it can have an impact on malaria. So that being said now, so given that we know that in most places in Africa, so that we have insecticide resistance problem, meaning that you can apply your bed net and apply your IRS, you will kill maybe a good chunk of proportion of mosquitoes, but still you will be left, with some of leftover mosquitoes that, your conventional control measure cannot reduce. Now, I think if you want to combine these two tools together, this is exactly where the gene drive is going to work. So meaning that if the conventional tool kill mosquito to some at some level, and then you have a leftover that cannot be controlled by these conventional tools, meaning that even if your mosquitoes mate with these mosquitoes, given that they will have the resistance profile, and if they get in contact with this insecticide treated material, they're not going to be killed. So in this case, I think the two tools can cohabit in the same environment, but in places, I know that all these are going to be kind of a little bit costly. And if you want to apply both IRS, both LIN, and also the gene drive technology in all these places, it's going to be a chunk of money. So I think we have to look really strategically to see what can fit where. If you have places where, you have a very good profile of insecticide resistance reduced, that you feel that, BNet or IRS alone can do the job, you don't have, to go necessarily to there to add other intervention tools, because you will add to cost. But in places where, maybe accessibility is really extremely complicated, and in places also where insecticide resistance profile is really very high, even though, you have this intervention tool there, but they cannot eliminate, at least a high proportion of mosquitoes, I think that the gene drive is going to fit there really very well. And it might seem like a little bit counterintuitive. Well, I don't know how to have a good English, but I believe you understand what I mean.
Yeah,
yeah, exactly. So that if you release your mosquitoes and then the insecticide is out there already, your mosquitoes are going to be in contact with the insecticide, you kill them, and then how your gene drive is going to spread. But I think the two can cohabit in that sense that your insecticide-treated material will never be able, at least for what we can see now, be able, to kill all your mosquitoes in the places. So you will always have, some leftover mosquitoes that the gene drive can take care of.
Okay, so on that point, and I want to just try and stress a point here, in case this makes sense. Earlier, you guys were describing one characteristic of gene drive, which is that even if the number of mosquitoes initially released is very small, there's still a chance that it will spread. Is that accurate?
Yeah, absolutely. But
so does this mean, therefore, that even if you had an intervention like ITNs or IRS that is also killing mosquitoes, that the few that are left still have a chance to spread?
Exactly.
Okay, so this brings me to a second question. Burkina Faso has a lot of pesticide resistance. pyrethroid resistance in the first place.
Mhm.
When you release mosquitoes that carry gene drive, do you intend that they will also be pyrethroid resistant so that they do not easily get killed by bed nets? Or do you expect that they will be fresh from the lab susceptible mosquitoes?
Well, I mean, it's not our intention, to induce the insecticide resistance per se, in the mosquito that we will release. But keep in mind that, when you're working in the lab with genetically modified mosquitoes, these are mosquitoes that you have kept in the lab, some of them maybe more than 20 years. So you have a reduced polymorphism there, and then also the phenotypic variation of these mosquitoes is also going to be really extremely reduced. So you have no idea if you take these mosquitoes straight to the field and then release them, are they going to be fit enough, to be able to do the job for what they have been released? So this is the reason why it's extremely important, to try to take the genetic element that you are interested in and then put it into the genetic background of your local mosquito population. And because you are collecting these mosquitoes from the field, so they have also all the genetic background with whatever is already out there in the field that you are taking in the lab, you introduce by introduction, this genetic element into these mosquitoes, and then you bring them to the field and then you release. For several reasons. The first one is, I already said, to make sure that, these mosquitoes are going to be fit in the complex ecosystem in which you're going to release them. But the second element also is that you really want to make sure that you're not going to be conflictual, with people saying that, oh, you're taking, completely different genetic element somewhere else, and then you're introducing that into our environment.
Okay, so if you come to my village, you will not be bringing any new mosquitoes. What you're saying is you will come to my village, you will collect some mosquitoes from my village, you will put the drive inside them, and you will re-release those mosquitoes.
Exactly. At least at the beginning, this is what is happening. Like, for example, if we get any genetically modified mosquitoes from our collaborator coming to Burkina, we don't take them straight to the field. So we have to collect, local mosquitoes, introduce the gene of interest into the genetic background of these local mosquitoes, and then release them. But now the question would be, do we have to do that, for every single country or for every single villages, even in Burkina Faso? But luckily, if you look at the genetic profile of mosquitoes, basically across large geographic area, you will realize that mosquitoes here in Burkina are similar to what you can find in Mali, similar to what you can find to some extent, at least in Cote d'Ivoire. So that's a really, very good thing. So it's meaning that if there is a regulatory pathway for these people, to coordinate this thing, mosquitoes, from one country might represent, the variation, phenotypic variation that you can find in many other places beyond this single country.
Thank you very much. And we will be talking a lot about regulations as well. I'm hoping that Dr. Willie Tonui has joined us. If you've joined us, Willie, please turn on your video and let us welcome you. But before we do that, I would like us to go back a little bit to molecular biology, and I want to talk to Austin here. So Austin and Tony, actually, either of you can take this. I would like us to go through a short sequence of description of the gene drive development in your laboratories. I know that you love double sex, but before we speak to double sex, talk to us about this initial description of the 3 gene sequences that you selected and why this particular one, number 280 at the end, was special, why you selected to go for this. This is, I think, the first description of a suppression drive from Imperial, I believe. Yeah.
Okay. I'm happy to do that. But if you don't mind,
yes, please.
Because based on something Diabate said, could you just go back to the other slide that shows the copying? There.
This one? Mm-hmm.
Yeah. So what Diabate was talking about there was the protocol at the moment is to integrate the gene drive into a local strain or a strain that is colonized from that country of intended release. And there's one important feature to consider with this type of gene drives, at least, is that in the process of copying the gene drive from the chromosome that it's on onto the new chromosome when it mates with a local wild-type mosquito, it's only the gene drive that gets copied across and introgresses into that chromosome. So it rapidly sheds its existing genetic background. And so it rapidly introgresses and integrates, so camouflages itself inside the locally adapted genetic background. And that's a quite important point with these gene drives. So once they get in, they have a hook, and then they're inside the local genomic context.
Okay. And when you say only the gene drive gets in, you mean only the Cas and the guide RNA?
Right, or whatever gene drive it is. But yes, in this case, more or less just the Cas and the guide RNA that copies across, and a few, maybe some small bits of neighboring sequence, but not very much.
Thank you very much. Let's proceed to this then. Take us through this and how you ended up selecting these specific sequences for your first suppression drive.
Okay. So in order to spread something that's designed to debilitate the population, you need individuals that carry a copy of the gene drive, just one copy of the gene drive. They need to be functional and viable. If they weren't, the gene drive would be a dead end. As soon as it found itself in an individual, those individuals, if they weren't viable, they wouldn't be able to transmit it to any offspring. Okay, so that means that when you have a gene drive, if you're designing it to disrupt a mosquito gene, an individual that has one copy of the gene disrupted by the gene drive and an ordinary normal copy of the gene should be viable. So that means you need target genes that when you disrupt them, just one copy of the gene, they're okay. And so you make a bioinformatic search considering a number of different criteria and try and work out which genes would fit those criteria, i.e., if you disrupt one copy of it, are the mosquitoes still okay? But when you disrupt two copies of it, are the mosquitoes then non-viable? That's what you want. Okay. And
is this easy to find? Are there, my understanding of genetics is that you have a lot of traits that are expressed in multiple locations.
Yeah. Well, actually, this is the first time anyone's actually gone and probed me on this, and I'm glad for it, because it is. You're right. It's a hell of a lot of work, but it gets lost actually in people getting excited about the copying. So the actual choice of target gene is tricky. You're absolutely right. And so with Drew Hammond, who's on the call, I think, we came up with a list of genes that we thought if we disrupt these in one copy, they would be okay.
That's what you have here.
Right. So there's 3 different genes there. All of them are, we thought, with roles in female reproduction. So the thinking was that if you disrupt both copies of those in the female, she would be non-viable, non-fertile. Whereas if those genes are not required in the male, even if they have both copies disrupted, they should still be viable because the gene is not required in the male. That was the thinking. And so what we first did was use these genome editing techniques to specifically disrupt those genes and see when one copy was disrupted, was it viable? And when both copies were disrupted, were they non-viable in the females? And that's what we showed. Okay, so that means that those genes fit the characteristics as target genes to be disrupted by gene drive. Okay, and then so when you actually place an active gene drive element within those genes, and you see whether it can copy from one chromosome to the other, it copies very efficiently, and it can spread in a population, despite the fact that it's disrupting something that's essential for the females.
And so I want to preempt a question here, but we're going to ask you again. This is not double sex.
This is not double sex, no.
Okay, good. So,
but double sex fits a similar set of criteria in the sense that when you have one copy of the double sex gene disrupted, your males and females are okay. And when you have both copies of the double sex, or the particular part of that double sex disrupted, The males are okay, but the females are non-viable.
Okay.
So the concept is essentially identical.
So around the same time that you guys described this CRISPR-Cas-based gene drive and Anopheles gambiae, a group in the US described an alternative approach on Anopheles stephensii. And I believe we have a diagrammatic for this. This is from Tony James' lab. And they worked with Anopheles stephensii. They described a different system that does not end in a population suppression, but rather just modifies the mosquitoes so that they no longer can transmit the pathogen. Now, I want to use this to ask a question about the drive itself. So first of all, just for clarification for my colleagues on the line, this is population modification as opposed to population suppression that target malaria works. Now, reading this paper, I see something that forces me to ask the experts. The sequences that they use here, they seem pretty huge. They seem quite a lot of payload there.
Okay. So
is this, do you have a limit in terms of gene drives? And either of you guys can answer this. Do you have a limit in terms of how much material you can put in these sequences for them to drive?
So this approach here, as you alluded to, is say a modification approach, but the principle of the copying, the way that the gene drive increases in frequency, is identical. It cuts the chromosome, and the whole thing gets copied across. As you say, there is a payload in here, which is in the form of expressed monoclonal antibodies that are designed to interfere with the parasite development. And so there are a few genes within this construct, and that whole construct, I can't actually remember, but it's relatively large. It's about 12,000 bases.
That's huge, yeah.
And notwithstanding it has a few genes in it and is a reasonable sized region, it still copies across at high frequency. And we've shown similar efficiencies in our labs. So at least to that size of construct, there doesn't seem to be any obvious reduction in the capacity of it to copy across.
Okay.
Of course, at some point there will be a limit, and it will decrease in frequency when you start to get very large.
Thank you. Another thing that came out of this paper, forgive me for going detail on this, was the question of whether it matters that you're using a male or a female for your initial copies. For the sake of the modification.
Well, the idea eventually is that this process of copying and the biased inheritance happens at the same rate in males and females. In most release case scenarios, there would be a preference to release only males, because you don't want to release a large number of biting females, even if it's only for nuisance. So usually there's a preference to release only males. But in terms of the initiation of the spread, it doesn't actually matter. I mean, Austin might want to comment further.
Thank you. Thank you. We're going to move forward, and maybe either Austin or yourself can comment on this. But we're going to come back to you very, very soon on this. Let's talk a little bit about your holy grail. Shall I call it your most prized gene drive product? Is that accurate?
I personally don't think it's helpful to call it things like Holy Grail, because it sounds like a trophy, as with more merit than other vector control approaches, which I don't want to go down that route.
But it is fairly unique and fairly powerful, right?
This is the best performing gene drive that we have. Yes.
Exactly. This is the phraseology that I was looking for. Your best performing gene drive. Please, Austin, describe a double sex in the simplest language you can. And I'm saying this, you're muted, Austin.
Right, I think I'm unmuted now.
Yeah.
Yeah. Okay, so Well, the most essential features of it are that it's like the previous genes in that if you knock out one copy of it in females, the females are fine. And if you knock out both copies, then the females are unable to reproduce. And the males, if you knock out one copy or two copies, it doesn't seem to matter. And I should say that's not a feature of the whole gene, it's a specific region in the gene that's only needed in females. So that's the most essential part about this gene, is that it's got these features of the previous genes. It's got the additional nice feature that the homozyte, the females who have both copies knocked out, not only are they unable to reproduce, not only are they sterile, they're also unable to bite people and therefore unable to transmit the disease. So that gives an extra bonus, if you want, in the control. It's not a necessary part of the control, but it gives an extra efficacy. Then the third nice thing about this gene is that, or at least this region that is focused on in this paper, is that changes in the sequence are likely to disrupt the function. So just like with any kind of pest control, whether you have to worry about the potential for resistance to evolve. So if you're dealing with chemicals, insecticides, then you have to worry about insecticide resistance. If you're trying to develop drugs, you have to worry about drug resistance. So, and similarly, we have to worry about resistance to the gene drive. And the most obvious type of resistance that could arise is some change in the sequence recognized by the enzyme, by your CRISPR, such that it's no longer recognized by the enzyme. And then, I'm not sure why we've changed the slide here.
I just pushed that up because you wanted to talk about resistance, so I thought that slide might help.
Yeah, well, I went.
Maybe not. Okay.
Go back.
Okay. Go ahead. Okay.
So if you look there on the right-hand panel there, the second part down, you see that the sequence is relatively well conserved within the Anopheles gambiae species complex, so that you get the same sequence. There's not many changes.
This is the blue mark here.
Yeah, so the blue plus the gray beside it, the gray to the left of it, that's the recognition sequence for this particular gene drive that is a construct described in this paper. So that's the recognition sequence. And you can see that blue part plus the underlined part beside it, that's conserved throughout the species complex. Yeah, so all those 20 or 21 base pairs are conserved pretty much throughout this. So that means that, or that was a clue that changes are probably not well tolerated in that region. And so that's the third nice thing about this gene, is that, and you can see for those who know some molecular biology, it's right at the junction between an intron and an exon. And so that's a region where this precise nucleotide sequence is likely to be important. And if you disrupt it, then that's likely to disrupt the splicing function. So yeah, that's those are some of the reasons that we're particularly got us interested in looking at this double sex gene.
So one question that we've been asked a lot, and we're going to bring this at some point to one of you, is about this as well. The question that you are working on one species, but Africa has so many species. What's going to happen? So let's hold that thought there. But first of all, an important question that has been raised here is, what if this thing fails? So a lot of my colleagues in the continent are asking, are you sure this thing will work? Okay, are you sure that the gene drive constructs will work? And Austin, you were alluding to the issue of resistance to the drive as opposed to insecticide resistance, but resistance to the drive. Could one of you use this or any other thing that you want to use to explain some of the limitations that we have with gene drive and why there's a chance that why failure is still a chance, an option, and a possible outcome?
Developing anything, you always have to worry about how it might fail, why it might fail, and try and minimize that likelihood. That's nothing unique to gene drive. So for population suppression, like with targeting the double sex gene or a female fertility gene, one has to worry about the potential evolution of resistance, target site, and particularly target site resistance, changes in the target site that might evolve. For population replacement, there is that issue to some extent, and there's a second issue. So the population replacement or population alteration approach, where you're trying to use gene drive to spread a genetic construct through the population, that a gene, for example, that reduces its ability to transmit the pathogen. The additional issue there is that at some point, some of the times during this copying process, you might lose that cargo gene, that effector gene, the gene that is responsible for stopping the pathogen from developing within the mosquito. And that if that in most designs, that would have no effect on the ability of the thing to spread, and it might even speed up the spread. And so you might end up spreading through a population a gene drive that has lost its cargo. And so that's an issue that needs to be thought through. And this paper then was trying to do some modeling, mathematical modeling, to assess how much of a risk this was, or what sorts of criteria would need to meet, molecular criteria would need to meet in order to avoid this problem.
So not all changes in the genetic sequence are bad in terms of functionality. You could have a mutation there, but it still works.
Absolutely, yes, that's absolutely correct. So we know many changes where a gene drive might be targeting might not have any effect and still be perfectly functional for the mosquito, but no longer recognized. So that's a concern for us, and we pay attention to trying to deal with that.
Right. Yes, Tony, go ahead.
So I think just with talking about gene drive, sometimes we tend to go from one extreme to the other. So at the start, we were talking almost as if it were a silver bullet. Mm-hmm. This is the great white hope, and downplaying the proven effects of vector control using conventional methods such as insecticides and habitat management. So on the one hand, it feels very unfair to sort of downgrade those methods that are proven, and everyone and loads of people on this call work on those, and they are absolutely proven, and throw it away for the fancy looking thing with the potential and say it's all on that, on the one hand. Then on the other hand, then the discussion moves from, why bother investing in this technology that has its potential? Yeah, fine, but it could very well not work. Do you get me? You've one flip from one extreme to the other. So, the fact
that's a question that a lot of my colleagues ask.
Exactly, and it's valid. It's absolutely valid. But antibiotics have had huge impacts on human health for the good.
Correct.
Yet all of them are eventually compromised by insecticide resistance, antibiotic resistance. The same for insecticides. It doesn't mean it's not worth investing in those technologies because of all the gains we get in the meantime, and the incremental learning that we get that allows us to, ameliorate those issues and plan and mitigate against resistance arising where possible. So I think that's a very important message. And, it's not stretching the analogy too far to think of some of these gene drives. This concept of targeting conserved regions is very analogous to how you might design an antibiotic. If you were doing so, you would try and target something in the bacteria that is very conserved and cannot freely mutate away to be resistant to the antibiotic that binds it, while at the same time doing what it's supposed to do in that cell. And so the concept is very similar for these.
Right. Right. No, thanks a lot, Tony, for that. And actually, if you don't mind, I would like you to carry on to also respond to this question here. With the diagram you have on the left side here, you have, if it was the best case scenario, this drive would just go straight to the top. But there's possibility that you have that resistance, and therefore it doesn't go to 100% frequency, and at some point it does. A common question has been whether this is actually a good thing for gene drives. Whether it is actually a good thing for us to go for something that we know will eventually disappear or eventually die down. Is this something that is in your minds when you do this work?
That there can be different outcomes, and that in certain situations the preferability of a particular outcome can be different. Yes, is true. I mean, perhaps Austin's better talking about that.
Yeah.
He doesn't mind me passing the buck.
Go ahead, Austin.
Most of the time we do try and think about ways to reduce the likelihood of resistance evolving, just because that's how we think, we think of resistance being a bad thing and we want to avoid it. And we have proven it's easy for, if you don't think about resistance, or if you want resistance to evolve, then that's easy. That's, we've proven that, in other words, right? So, so we spend most of our time thinking about how to reduce the likelihood of it arising. Yeah, I'll just leave it at that.
Okay, let's talk to Dr. Diabate a little bit. Diabate, in Burkina, can you hear me, Diabate? Brilliant.
I'm on mute. Yes.
Yes. This is work also from Imperial College, and I want to put this together with work that is coming out of Anopheles gambiae 1000 genome, which I'm sure you participated in. And the question here is, in Burkina, you have Anopheles colutsei, you have Anopheles gambiae. You also have Anopheles funestus, you have, I believe, Anopheles arabiensis as well, and several other anophelids. Unlike in other parts, you have two vectors that contribute quite heavily. So Anopheles colutsei and Anopheles gambiae are both very good vectors. A common question, and I asked this earlier, is what happens if you're successful with one of them, but not the other? Or is your strategy in Burkina Faso to go after both gambiae and colutsei?
Okay, so we'll look at the question even beyond, the complex species of Anopheles gambiae, but as you already mentioned, in fact, you have other species like funestus, you even have Nila. They might not play, the same role in malaria transmission than, these other species. But one thing which is really, really extremely important, to keep in mind is that gene drive is not coming, as a bullet control tool, silver bullet control tool. So it's coming in complement of other intervention tools that are out there. Meaning that if you take, for example, a gene drive construct, and then you have it, let's say in Anopheles gambiae or in Anopheles colutsei, the beauty of it is that obviously, if you release this mosquito, it's going to target specifically Anopheles colutsei if the construct is in Anopheles colutsei. We obviously know, that we have reproductive barrier, within the complex of Anopheles gambiae, but this barrier are porous to some extent.
Yes.
So we believe that if you have your gene drive, for example, in Anopheles colutsei, which is already pretty much common all over the country, if you release and then it's working, there are chances that your specific construct can, introgress into Anopheles gambiae. For sure, because I mean, we have already shown that, you know, with the insecticide resistant, the KDR mutation that we have been studying, you know, for a long time, we found that it was in one species and then it has been progressed in the second one. So I believe that the gene drive also can follow the same path here. Right. Now, question might be, if you move out of, you know, Gambia and Colucci, what will happen in Arabiensis? Arabiensis up to now, basically, in Burkina Faso, we have Arabiensis, but the contribution of Arabiensis to malaria transmission is quite low compared, you know, to the two other species. And if you look also at the insecticide resistance profile of Anopheles arabiensis compared, you know, to the two other species, it's also really completely different. Right. For the simple reason that, you know, I mean, they don't have the same natural exposure to this chemical into the environment that can, you know, select for the resistant trait. So the hope here is that gene drive can do the work for, you know, the major malaria vector. And then because you have other intervention tools that are out there that might help, you know, to tackle the other, like Arabiensis, like Funestis, or even Nila. But beyond all this, I think that also it's a technology which is adaptable. The first step, we're testing it, you know, with Colucci or Gambia. If it's work, and we have a, you know, very good understanding of how it's performed, it's can be really quickly adaptive, you know, to other species. The Arabiensis, it cannot progress to Arabiensis, or other species. The Funestis, I know yourself is working really, very hard on Funestis. We have like a very good, very strong bottleneck there because it's extremely difficult, you know, to rare Funestis and in the laboratory to be able to collaborate
we're losing you a little bit, Diabate.
Funestis in the lab.
Yeah. Oh. So just one point there. In this paper here, in this publication here, the group is, and I think actually that I saw Ness on the call join today. There is very good evidence here of some great similarities between the members of the Gambi SS, the blue line there. Of course, a bigger distance between this Anopheles and other species. I have also seen, Tony, from your lab, I believe actually we have that here, if I may just share it real quick, evidence of introgression of one of the gene drive systems that you have in Anopheles arabiensis in the lab. So I guess for us, the question here is how easy would this process, it appears from work done on the lab that it's a fairly straightforward thing, introgressing within members of the Anopheles gambis and Sulato. But the question is, how easy would this be to do beyond the laboratory? Is this a strategy that we want to assume is obvious, that, you know, once we develop something good for Gambi SS, we can just assume that it's introgressible into the other members of the Gambi SL?
Well, that's a good question. This slide you have up here is work done by Federica. I hope she's on the call, but it's slightly different in the sense that it depends where and what type of gene drive it is as to how likely you're going to get complete integration. Depends on those factors. It depends also on which of the two species that between which you're trying to introgress. And so in this example, she showed that you could get relatively good introgression of most of the genome into the new species quite quickly. Of course, this is under laboratory conditions, and this is either to open up the possibility of avoiding having to put the gene drive artificially into that strain first and then rear that strain and release it, or whether we could get around that by introgressing it, as well as considering what might happen if there are natural examples of hybridization and introgression. So it depends, but the message is that the likelihood of all these things happening depends very much on the gene drive and the target site that you choose, whether that's conserved between the two species in question, and whether they are reproductively isolated and to what extent they are, and whether they are co-occurring.
Okay.
So these are all factors that one would have to consider.
Thank you very much. On the question of conservancy or conservation, I don't know what exact terminology I should use here. I am wondering whether our colleagues from the Gambi 1000 genome program are on the line, but Tony, you can help me with this. There is a graph at the bottom right there.
Yeah.
So these guys have just recently published their second piece with this time with 1,142 mosquitoes, millions of variations they are seeing here. They were kind enough to actually zero in on the double sex region. I read this many times, but I failed to understand. In simple terms, did they find too much variation to be optimistic or too little variation to be optimistic? What is the finding here?
No, this is there isn't a strict cutoff, but what this is doing is using sequence conservation as a sort of a proxy for conservation of function and constrained regions that can't easily mutate. So if you consider that black bar, that gray bar there is part of an exon, so all of those DNA letters are converted into a message which creates a specific part of the protein. So there's a specific stretch of amino acids that form a functional region of the protein. And so if you've got a region of the protein that's absolutely key and cannot be tweaked or changed, then you would expect those amino acids never to change, because if they did, you would have a non-viable organism. Therefore, by extension, you expect the DNA that determines what those amino acids are not to change either. Okay?
So,
whereas, wait a second. So if you have another region of that same bit of the protein that wasn't so crucial, if you had changes there and they could be tolerated, then you might expect to see changes in the DNA that accumulate naturally over time. And so you would see less conservation of the DNA bases that were here and more conservation of the DNA bases that were here at the crucial region. And so by looking at the regions that are conserved at the DNA level between very different Anopheles mosquitoes, that will tell you that the region of the protein encoded by that stretch of DNA is probably very constrained, i.e. cannot easily be changed. And that makes it, again, with this analogy of the antibiotic, a good place to target, because you won't be able to easily change away.
So just simplify this again for clarity, and I apologize to everyone like me on the call. The subject we are handling today is a pretty difficult one, so I'm hopeful and I'm grateful that Diabate, Austin, and Tony are really helping us with the simplification here. Tony, the graph, or Austin or Diabate, the figure at the top right, the one that has clusters, this is also from the Gambi 1000 genome. It shows some differentiation between mosquitoes from different countries. Now, when you were talking earlier, you were very specific that this thing depends on a specific sequence, not necessarily the general differences in gene flow, the patterns. When you look at this data and these findings such as this, and it would be great if we have colleagues from Gambi 1000 in here who want to chime in on this. When you see this data as a whole, what does it tell us about feasibility of gene drive approaches across in different countries? Diabate was saying earlier that, mosquitoes in Burkina might be the same as mosquito from Cote d'Ivoire and all that. What are the chances that a single release in one country will have transboundary movement and successfully establish in another country, just based on evidence we have today? Is this a regulatory concern? And how does that match with the data that we have now, especially from the Gambi 1000 crew? Does that question make sense?
If I may, I think, so the data is showing that there are recognizable or distinguishable differences between the mosquitoes in the different countries where they've been collected.
Mm-hmm.
So Africa is not one great big mixing bowl for Anopheles gambiae. This data is from the full, basically the full genome of the mosquitoes. So huge amounts of data has gone into this, and using huge amounts of data, you can find distinguishable differences between mosquitoes in different locations. And the amount of those differences does tell us something about how much movement there is from one place to another. But it's still, and if you look at the middle graph, for example, yes, exactly that one, that's the data at the moment is suggesting that, let me get this the right way around, that Gambiae is moving more than Colucii. Yes. That's what the data is suggesting at the moment. The Gambiae, the flat, in the middle there, the line is flatter for Gambiae than for Colucii. But precisely how we can translate this genetic data into estimates of how much movement there is, that's still a topic of study, not just for mosquito biologists, but for population geneticists at large, the whole field, how to be able to read and interpret DNA sequences in a way that we can learn things that we care about, like how much they move around.
Okay.
And so that's an ongoing bit of activity. And so I don't think we can give a precise answer at the moment about the rate at which a gene drive released in one country might spread to a neighboring country, but that's something that we and others are trying to work through.
Okay, so this is an important concern. Diabate, you are muted. You can unmute. This is an important concern that some countries say, if my neighbors release the drive, I need to know. And so this is why for us, it's like, based on all the molecular data that we have today, for example, from places like Gambi 1000, from the sequences we have, what are the indications that this is even a possibility? But I get your point, Austin. Diabate, go ahead, please.
No, I just wanted to have a little comment on how this thing is going to spread. Obviously, I mean, it's a really, really complex ecosystem environment that if you release these mosquitoes, you have absolutely not a clear 100% understanding of how fast it's going to, move across landscape. But what I wanted to say is that, as Austin already mentioned, if you look at these differences, this is really at the whole genome level. So even in a single country, if you look, depending on a specific ecological zone where you collected these mosquitoes, if you look at the entire genome of these mosquitoes, basically you will see that you have a specific, I don't know how to say that in good English, package of mosquitoes that are really kind of a little bit different. Yeah. What I want to is that to reflect a little bit on the insecticide resistance KDR mutation thing. Obviously, if you look at the history of KDR mutation, they might have, several, at some point, some multiple origin of the KDR mutation, but most of the studies really kind of showing that you have a single origin. Like if you have the KDR mutation, for example, emerging in one country, let's say, I think that for the KDR mutation, at least in the beginning, my understanding was it has been detected in the first place in Cotonou, in Benin. And then look at the KDR mutation today. So it's all over the places. To go anywhere in West Africa, so you have KDR mutation. And we believe that at least for those specific species where at least the reproductive barrier is porous, that the gene drive is also going, to follow the same path. It may take some time. I don't know how long it's going to take, but at least if you release, you might have some specificity here and there, but the very important component that can allow this gene, to move into the different population across landscape in one country or to the other country. Basically, we have already example with the KDR mutation, and then we believe that probably it's going to be the same as well.
Okay. Thank you so much, colleagues. Thanks, Austin. Thanks, Diabate, and thanks, Tony. I would like to ask Delphine or Kairos or Charlotte, if you guys have any questions that need to be answered by our experts.
Yeah, there are many.
Could you select one or two before we proceed?
Okay, I'll start with some of the first one we had. There's been quite a lot of questions around ecological impact, how you would assess them, and with this like behavior of the mosquitoes, yeah, how you would assess those ecological impact, what happens if you suppress mosquitoes. So maybe we can start with this.
Cool.
Yeah, I'll start not by fully answering the question. I'll leave that perhaps to Austin or Diabate, but it's an observation that gene drives are not the only thing that are trying to suppress populations of mosquitoes. As we said before, there's been huge success in reducing the malaria burden by targeting mosquito populations and suppressing them, either by removing their habitats or by the indoor residual spraying, or more latterly and more successfully with the insecticide-treated bed nets. And so it's important to put it into the context of other suppressive vector control methods is my message. And while it's a valid concern, it should be applied across all vector control methods.
Who is speaking here?
Kentega. We're having Kentega who should mute.
Kentega mute, or I remove you from here.
Yeah, I think that there are two questions in the question of Austin. If I say Austin and Delphine, if I understood it really, really correctly, how are these mosquitoes going to behave in the field? And then what will be the ecological impact? This is what I understood. But I think these are two questions. So the first question aspect is that how are they going to behave? And this is something that is extremely important. Again, remember, we said that you are taking this mosquito from the lab. They have been adapted in the laboratory condition, for years and years. And then you transform them. You want to take them to the field and you want to release them. It's clear that they have reduced polymorphism, and then the phenotypic variability also is going to be reduced. So we don't know if they are going to be fit enough in the natural environment, to operate as we want them, to operate. And you have a chunk of studies that a lot of people are working on, either in the lab or in the semi-field screen that mimic, the natural environment, trying to understand also, the behavior of these mosquitoes that have been kept in the lab, and how can you optimize, these by backcrossing, to other mosquitoes that you collect from the field and then optimize this phenotype. So there are a lot of studies on that, be on the survival, the fecundity, or even the mating competitiveness. All these aspects basically are part of the pathway of, development of the technology. Now, the second question, let's say that you release this mosquito in the field and then it's working really perfectly. For example, you're targeting Anopheles gambiae or Anopheles coluxi. So what impact is going to have? Are you going to remove them completely from the environment? And if you remove them completely, what is going to be the impact? So on this question, I think that we don't have 100% answer to say that here is what's going to happen. But for one thing sure, exactly what we are doing now is that we have a number of studies that we are conducting, not only in Burkina, but in all other countries that are involved in target malaria in Africa, looking at the impact of, the release of the gene drive. When we get there, obviously, what will be the impact, not only on the non-target organism, but also, on the sibling species that have not been really targeted specifically. So one thing also important is that to keep in mind that take, for example, the insecticide that we are using. I know that insecticides, for example, did not have the biggest impact in West Africa compared, for example, to East Africa, sorry. If you look at the data from East Africa, so you have the impact of these insecticides basically wiping up to some extent, not completely, but at least to some extent, reducing very drastically, the density of some of these mosquitoes, Anopheles gambiae, in the point that, some of these mosquitoes are really very rare. And then you have now Arabiensis that people seeing pretty much everywhere, and also Finensis contributing a little bit more, to the resistance. To malaria transmission. But to my knowledge, and you are from East Africa, you can correct if I'm wrong, do we have any specific ecological serious impact that have been noted, because of the insecticide that have been applied in Bennett or IRS, are kind of wiping out a little bit less Gambia? I'm not aware of any possible, but we have to take time really, to study all these different elements to make sure that if we say we are ready to release these mosquitoes in the field, we have taken really time to look at all these different critical elements to be sure that we can bring clear and solid answer to the regulator before we can do it. Yeah, not sure what was the second question, but anyway, so I'm going to leave this to Austin if you have anything to add.
That's fine. Delphine, you have another question. You said you had 2 questions.
Yeah, I have more than 2. There's many more.
we can take 2 for now and then.
Yeah, there's a lot of questions around vector competency. Would the gene drive be able to transmit malaria in a better way, or new disease, or become a better vector or vector of other things? And I know there's a lot of research on that, so maybe if you guys could say a few words on it.
Yeah, that's something that obviously we need to be concerned about, and and to a certain extent, that's a difference then between suppression approaches and alteration or replacement approaches. If you're substantially suppressing the numbers of something, well, it would be difficult for it to transmit to any significant degree. But anyway, nonetheless, it's important to do those studies to look, and so those can be done in the lab to check whether the vector competence, the ability of the vector of the modified gene drive mosquito, have we somehow changed inadvertently its ability to transmit malaria or a virus, for example. And so that sort of experiment would need to be done before any releases were done. And we have done that sort of experiment so I'm not sure if we're going to, at some point, probably we'll get to the fact that in Burkina Faso, there was a very small scale release of non-gene driving genetically modified mosquitoes, sterile males. And those sorts of tests were done prior to any kind of release. And certainly one would want to do those tests again on a gene drive strain as well.
Thank you. So, Tony, are you still with us?
Yep.
Oh, okay. Delphine, feel free to stay on video. We actually have questions on engagement that you might want to answer. But let's move a little bit to the issue of containment, and it would be nice to hear from our experts about this very, very briefly. In target malaria, at least, you guys are starting to move towards eventual field deployment. You haven't done that yet, but you're moving towards that. And so one of the things that people start to ask is, what is going to happen when you are preparing? How do you take care of these organisms? And a simple question for you here is, do you need any special containment measures for a gene-drive mosquito? And if it is it arthropod containment level 1, 2, 3? Is there anything?
Yeah, well, Tony, you want to go? Or
no, go ahead.
no, what I wanted to refer to, Fredros, I don't know if this is, I wanted to add, to reflect a little bit, on the pathway of, testing the gene drive, because, I mean, all these different elements, in fact, are contained in this pathway that we will have.
I'll put up a slide for you for pathway if you want. Yeah,
okay. So the thing that, talking of the pathway, I must say that you have different pathways that have been, proposed here and there by different group of organizations coming together. You have this different pathway out there, but there is no set on stone every pathway that is already there, that all the scientists know that, okay, this is the pathway really that we need to follow. That's the first element. And then the second element also is that all the different pathways that you already have out there are derived to some extent, you know, from an original pathway that was drafted, you know, by the WHO long time ago with, you know, simple genetically modified mosquitoes. It does not have, you know, the gene-drive construct. And then they revised it at some point. But
this is the one we have here. This is the 2014 one.
Exactly. So, but the content of this is what? If you look at this pathway, what is clearly showing that if you start with, for example, the phase 1, this is something obviously that you need to do at the laboratory level. You know, I'm just talking about the genetically modified simple mosquito with no gene drive, so that we can make the connection later on, you know, the gene drive itself. So you start with the phase 1. This is completely in the laboratory, cage studies, and it can also even be large cases, but inside. The reason for that basically is that because you are at the beginning of the development of this process and you really don't know what is going to be the efficacy, but also the safety issue in the natural environment, you really need to start with something simple where you can control the spread of your mosquito. So in the lab, if you do this kind of study, what you are interested in is obviously the safety issue that you have to start looking at, but you're looking mostly, I would say, the biological and functional characteristic in a really very controlled environment. So once you have clear data there, and then you can take this construct now, you know, to the next step. In this model that you have from WHO with the genetically modified simple mosquitoes, the next step was the phase 2. And in the phase 2, what you do basically is that you use these mosquitoes, I mean, in a kind of confined environment or physically confined environment. The principle is still the same here, because you really don't know what is going to be the real impact in the safety, at the safety level of the thing. So you still have to make sure that it's really, even if your environment is kind of a little bit close to the natural environment, you have, you know, physical or ecological containment, that confinement that will allow you, you know, to control the spread of your mosquitoes. And there, what you look at is, I would say, you know, still the biological and then functional characteristic, but in interaction, you know, with wild mosquito. Right. When you pass all these elements and then you have satisfactory answer, both, you know, in the biological, the efficacy and then the safety, then you are entitled, you know, to take this mosquito now to an open field release. It can be at various levels, small or bigger field release, and then you follow up. This is the phase 3, and then you go to the phase 4. But the particularity with the gene drive here is that there are some modeling showing that even if you have few mosquitoes that escape and that are in the environment, then the gene is going just to go to spread. And then it will spread and then it establishes itself, you know, in the population. So it's really extremely important in this case to say, for example, the phase 1, we all understand that this is done in a really very strict, close containment, so that you have a absolutely control, you know, on the spread of your construct. But moving from there, you know, now to the field, with the ecological containment or the physical containment or even the open containment, I mean, the line between, you know, these different type of containment is not as clear with the gene drive as, like, you know, with the conventional genetically modified mosquitoes. But in all, what is really very important here is that the first phase will happen really in an environment which is really completely closed, where you have absolutely control on the spread of your gene drive. And now when you move to the next step, you have to make sure that you have all the specific data related to the safety issue that you can bring to the regulator, showing them that, I mean, you have think of any possible test, I don't know if this is good English, but anyway, testable hypotheses that you have clear answer that you bring into them, and it's up to them now, you know, to look at this data and then to decide if they will give you the authorization, you know, to live in an open environment or not.
So Diabate, in your specific case, and feel free, guys, to stay on this slide if you want, because I think it's really important. And again, I'm asking if Dr. Willie Tonui is joined us. Kindly turn up, because we've got lots of questions for you. In this specific case of Burkina Faso, do you have, for your small scale initial work, do you have any special requirements that you want to achieve before you proceed?
Yeah. First of all, I mean,
is it ACL 3 or 2, or does it even matter?
Yeah, well, we have ACL 2, and then when we move to gene drive, so it's mentioned that we may have ACL 2 plus and something. But look first at the pathway that we have put in place.
Yes.
We have not started straight with the gene drive construct, and that is really extremely important. So we have started with a sterile male self-limiting construct, and that allows us to do both the laboratory and then the field small-scale field release that we have done. But this allows us to have all the information, but at the same time to have a really very good control on the spread of this genetic element that we are introducing into the environment. So this is the one really step very important. And then the next step that we are going to move on now is a second step, whereby I know it's not a sterile male construct, but it's going to be a self-limiting still. The mosquitoes can spread, but it cannot spread as the gene drive. And then, so it means still that you have control over the spread of the transgene that you're going to put into the environment. And that will allow you, to look at all these different elements and think, well, okay, do I have enough data, both on the safety, but also, the efficacy aspect, just to allow me now to take this same construct and put the gene drive in it and then go to the field and then start releasing it. So this is exactly what we have done. And this is to some extent what is reflected also on the pathway here that you can see. But at least in the case of Burkina, we have not started straight the gene drive itself. So we're going step by step, making sure that all the different concerns safety-wise that people have that can be addressed reasonably and in a really very controlled manner, so that the construct is not going to spread beyond what we are expecting, are completely under control before we.
So Dr. Diabate, I'm going to ask you to stay on that specific one. Sorry, where are we? I believe this is what you're referring to. And I read this article from Mark Benedict a while back, and the suggestion that for the transgenics, when you first go to the field, it's best to do this sterile insect, to do this the sterile releases. And I think this is what you're describing. Does it mean, therefore, that because you guys are the first in Africa to do this, that your journey is not necessarily on that WHO pathway as defined, that you have certain parts of that path that are just for learning purposes? Is that correct?
Absolutely. I will say, both WHO and all the different organizations, because gene drive have never been released in the field. So everybody is really learning in along the pathway. So, I mean, a concrete specific every pathway will come out, from the experience of the different team.
Okay.
But still, it's really very important. I think the most important thing is that scientists or the technology developer have, to be responsible, of what they are taking and then putting into the environment, and making sure that they have a really very good understanding of what can be the consequences if something goes wrong, and how can you really prepare yourself, to bring specific answers so that people can feel that, oh, we are confident now with this data that you are bringing to us, for us, to allow this to go into the environment, be ourselves or be anyone else. But the path is going to depend on the specific construct that each of us is developing. And as I said, also, it's going to be a constructive contribution from the different groups, to have a clear pathway for everybody.
Okay, thank you very much. So to Tony and Austin, this is an important point that the Diabate is raising here. If you look at the path they suggested this is the WHO pathway. I know that it's under revision, and soon we will have maybe a slightly modified version. This is the pathway that consensus based that a number of scientists worked on a few years ago. What Diabate is saying is that when you are the first, in their specific case, they are doing a lot of learnings, and so they're not necessarily following this strictly the way it is. From a very general target malaria perspective, or from Tony's perspective, do you expect that every group will follow the same path as Diabate, or they will reach a stage where we have learned enough that we then come back to this path and just stick to it? Maybe Tony or Austin.
Well, I'll point out that those WHO guidelines on the left are from 2014, when the first proof of principle of gene drive and its feasibility was still a hypothetical. So the one on the right are not too dissimilar, but are, say, more concrete, with a frame of reference specifically towards gene drive. Yep. So I wouldn't consider it one or the other. I think one's an evolution, if you like, of the other. And I think that being it's such a new field, there is a lot of discussion on the best way to get a standardized pathway for testing, what needs to be accompanying that in terms of public engagement, communication of the science, how best to do that is all still being a little bit worked out. And I suppose target malaria, who I should say I used to work for directly, are sort of pioneers there, because there isn't an existing trodden path down which to go. And so, from my perspective, as someone that works on largely the technical side, or at least had done until quite recently, only on the technical side, it's eye-opening for me to see how much other work is involved in terms of actually trying to get this to a functional product. And the technical testing and the gene jockey stuff and all the rest of it is only actually a fraction. And Austin will comment more, but I hope maybe for the rest of this call, some of the focus will be on that, because I think that those things are at least as challenging as the technical side.
Good. So Austin,
I'm not sure I have much more to say, just to reiterate that I think the intention of these documents is that they're living documents, and everybody expects them to evolve, as indeed they have done. Right. And I'll learn about this whole new platform for malaria control.
Right. So there is a group of scientists who believe we actually need even more stringent controls. And I have a slide here from, I believe this is Omar Akbari's group. And a clear suggestion they have made here is that we need to combine different approaches of containing this. I mean, if you speak to Kevin Esveld in particular, these guys are really pro tough controls on gene drives. The question to yourselves as experts in this field and pioneers in one way or another is, how feasible is it once you are out of the laboratory to combine more than one or two of these, more than one of these things, put any of these things together? They are talking about you need to have a physical barrier, ecological isolation, and with a strong idea that human error is common and you need not to rely on it.
So I think these recommendations they're specifically for experiments in the laboratory. Mm-hmm. So they're not really meant to be addressing once you leave the laboratory.
Okay.
So you know, and the intention would be that a great deal of safety work would be needed in the laboratory before you would move out of the laboratory. But these recommendations were specifically around laboratory-based research.
Cool.
Does that make sense?
Okay.
Yeah. Diabate, you want to say something?
Oh yeah, well, what I want to say is that obviously, I mean, we really need a very good, you know, confinement strategy to make sure that, you know, we're really moving on a safe path. But at the same time, I think it's also really very important, you know, to recognize that we don't want, you know, to put too much stringent regulation that is going to kill the development of technology. That has really to be kept in mind. We're all aware that, I mean, what we are developing, it has a potential, and we also know that we are really accountable and have to be transparent with what we are developing. Now, collectively now, how can we really discuss, because if you look at the different elements that are there in the table, even if you were taking this, you know, into the field, so there are possibilities, you know, to combine some of these things depending on the specific location where you are. But it's clear that it's not just the physical or molecular aspect that is really very important, you know, in this confinement strategy. You have all the, you know, regulatory issue element that you have also that's going to play in, the public acceptance element also that are going to play in, and all these are elements basically that are going to play here. So if you want, you know, to take all these different elements and then combine them, it might be really very difficult that you will find only one or few places where you will be able, you know, to combine all these things. Now, the real question is that what are really the most relevant research questions and the fear that we have, and given, you know, the landscape that we have, and we want, you know, this technology to develop, how do we really make sure that we can find a kind of very good combination between, you know, the technical aspect of the thing and also the regulatory and then the governance element put together, you know, to help us, you know, to move this technology forward. The elements that are there to me, I mean, are really very good elements, but we have to take into consideration other elements as well.
Thank you. Thank you very much. I think we should use the last few minutes left on this to discuss two things. One is public engagement. The other is I would like us to look at some of the data that is coming out of the modeling groups and discuss how this plays out in the field. And lastly, I would like us to discuss issues of capacity building for gene drive development in Africa specifically. So to begin with, let me put this up just for the discussion. Yes, so there is no data on gene drive to show that it works in the field. And I have to tell you, this is the first time I've seen a technology that people talk about so much, and yet there is zero data. It's the first time I've never seen any technology that is so widely spoken about without any field data. There must be a reason for that. And I mean, you look around, the only thing you find is some modeling studies that suggest that, you know, even if you release a few mosquitoes, it will work. And the first one you see there is from Philip Elkoff, one of the very early modeling assignments that demonstrated possibility, great feasibility of this. He also showed that actually, even with seasonality, the drives will still spread very well. You have data modeling work from ACE North, who I believe is on the call, also suggesting that in Burkina Faso specifically, that these things would spread regardless of you know, a few challenges here and there with estivation, migration, and all that. Now, in your own opinion, as experts working on this, how translatable is this modeling information to decision making? Even how do you use this information to convince us to keep moving forward, given you have zero data in the field?
Well, I think the modeling is essential on a number of grounds. It helps to direct the molecular biology in particular directions. For example, to emphasize, it helps justify that targeting female fertility is a better approach than targeting essential genes. It helps, and that resistance has to be dealt with, and these are the sort of criteria you would need to meet in order to reduce the likelihood that resistance will evolve. So I think it's essential for that, in sort of directing the molecular biology, and then also for extrapolating, the molecular biology. So when you say there's no data, there's wonderful data coming from the lab about
from the lab, yes,
for suppression of a population, and important numbers like the rate of the transmission and the rate of sterility and the various fitness effects and the rate of spread through a population. And so the role of the modeling there is to be able to extrapolate that data that you get from the lab in order to make a prediction or expectation of what would happen if this was released into the field. And so both these papers that you've got up here are attempts to do that kind of thing. And so, for example, one of the conclusions from the modeling is that, particularly from ACEs, that, for example, while in the lab we get populations going to elimination, that's probably unlikely over a whole landscape scale of thousands of kilometers, and that you would more likely just get substantial suppression but not complete elimination. That's our current understanding from the modeling of the likely expectation from this.
Tony, you have used modeling also to find possible resistance allele.
Not necessarily to find resistant alleles, but to model what impact those resistant alleles would have, and were they to arise at a given frequency, would they jeopardize the efficacy of a gene drive program? So those are important questions, and as Austin said, prioritizing which traits to affect in a population. One can get a lot of information there from modeling, but in a way, I mean, you're very right. The excitement that is attached to gene drive is not matched by the field data, but then it's a sort of a chicken and an egg situation, because the excitement that comes with it and its potential for being transformative also leads people to be reticent with some justification about, releasing it, because it's designed to be invasive. And this is a paradigm that's not been confronted before with a genetic control approach. There are biological control precedents, and there's perhaps the Wolbachia as precedent. But in terms of a genetic modification and spreading it into a population, that needs authorization, it needs in-country approval, community acceptance, and all those things. And that's what I was trying to allude to earlier. You need to get all of those on board in order to be able to ever get the field data. And so that's where a lot of the work is at the moment. And, that's what will allow us eventually to get the field data.
Right. Do you, and I really, I'm not sure if Willie eventually joined us. We really need his help here. Tony, you just mentioned the regulatory issues, and one of the questions that my colleagues have sent out is just which regulatory guidelines in the continent are relevant when it comes to this product. If you're saying that someone has to make an approval about that, you talk to these people, there's Cartagena Protocol, there's the Earth Charter, there's Convention of Biodiversity, Nagoya Protocol. Is there anybody in the audience who deals with this thing on a day-to-day basis, or maybe Diabate yourself?
Delphine has done a lot of work on this.
Yes, how does gene fit into this, and where do I start if I wanted to get my work in the right books?
Right books. Okay, I'll try to give it a go in a simple way. So there's two ways that gene drive for malaria get oversight, beside the whole obvious ethics protocol, like any research involving human beings. The first way is, of course, under the World Health Organization. You have the whole process around new tools, innovation, and so that's reviewing data around efficacy, safety, and so on. And this is more, it's not a regulatory per se. It is more an oversight to provide guidance to countries and provide a framework in which studies on entomological, epidemiological results can be done. So that, I think, people here would be more used to. The other framework, which people here might be less used to, is indeed the Convention on Biological Diversity, which is a UN convention that applies to the protection of biodiversity. And within this convention, there is a specific protocol called the Cartagena Protocol, which deals specifically with living modified organism. And so anything that is what we would usually call genetically modified organism, and for any release or movement of those, you need to follow specific regulation. And so that's a global framework. And then countries that are parties to it, that have signed that protocol, then have their own transcription of this in their own law. So I'm a country, like I'm Burkina Faso, I sign into the Cartagena Protocol, and then I will make a legislation in my own country on how we'll deal and implement those things. And so if we take the example of Burkina Faso, they have a biosafety law that explains how researchers who want to work with modified organisms would have to work. So there's provisions for when you want to work in containment in the labs. And so it tells you exactly how you would do it, etc. But it also tells you have to get a permit for it. So it tells you, there's a form that you have to fill. It's complex. It takes a lot of questions we had from the audience around risk assessment. You have to provide data about the fact you analyze this risk, etc. They review it, and then they give you an approval or not to work in containment. And the same way, they have a process for release, where still under the Cartagena Protocol, where they explain how any GM organism would be released. Again, you have a big application to fill with a lot of data to explain what are the potential risks, how you would control and mitigate those releases. And so in the last few meetings of the Convention on Biodiversity, it was clearly stated that gene drive is a living modified organism in the sense of this convention. So gene drive fits into this framework. There's no need for a new framework for this technology. Now the questions are, do we need specific, and this is a question we're hoping will be solved at the next meeting, which COVID provided should be by the end of this year, whether the risk assessment experts and regulatory agencies would need additional guidance on how to do a risk assessment for gene drive. Are there specific things in gene drive that need new guidance on how you would do this assessment? At this point, it seems that the guidance from the conversation, but again, the decision hasn't yet been negotiated, but it seems that this guidance would be additional to what we have. And so they wouldn't need to be finalized before you keep going with the research. So that's kind of where we are and where gene drive fits. In addition to this, in some countries, like in Burkina Faso or even Uganda and probably many other countries, there is a second aspect of oversight coming from the environmental ministry, which asks for environmental, social, economic, and health impact assessment, which is a complementary a process that you have to do where you look at both positive and negative impact, and you explain how you would mitigate those. And in very short, that's the framework we are in right now.
Thank you very, very much. Thanks for the clarification. That makes makes a lot of our work easy. And I hope that people understood. Anybody wants to add anything?
Yeah, I think I'm surprised, Frederick, you haven't mentioned your own work in this with Lena.
Yeah,
on the softer side, you know, and stuff that Target Malaria has also done about, you know, gathering opinion on perceptions of gene drive and what's involved, and, you know, what are the local sensitivities, because you get a huge difference if you ask local, say at the village level where there might be a release, to, you know, national scientific level, to national political level, or general cultural differences between, say, an African release scenario and Europe or America. And I think those are quite important. And, you know, I'm always amazed when I'm in Africa and talking about some of this stuff, and the temptation is to focus on the technical stuff like we have done with the genetics, and often the concerns are more like, well, how do you get something so bloody small into a mosquito embryo, into a mosquito egg? That thing is so small, you know, and that's the thing that they want clarity on. And so, you know, all audiences are different, you know, from the low level to the high level, and it's important to know what your local, I don't like to use the word, but sort of stakeholders' sensibilities are.
I mean, this is an important point, Tony, and you asked for Lena. There she is on the image. So.
Oh yes, I have the chat box over her. Yes, she is. Yes.
Yes. There she is, asking exactly the same questions, and I hope Lena is on the call, because the next question is for her actually. So there is this image here is from Gene Drives in the Horizon. This is the document that came out of the National Academies of Engineering, Mathematics, and Medicine in the US. And there's a way that they classify the so-called the publics, the stakeholders, and the community. Now, my colleague Lena and several other people in West Africa and Uganda have gone out there and really spoken to different people and asked, what are your opinions about this? In a recent example, they have asked people, what do you think? What do you know about gene drives? So, Lena, if you're on the call, I would like to request that you just turn up and explain this, because then I would like to ask our experts whether these are the same sentiments they pick up whenever they work in Burkina Faso, in Ghana, Uganda, and so on. Lena, please. Are you there? Can anyone confirm if Lena is on the call?
She was, so far she was putting things in the chat.
Oh, okay.
Yeah,
Or we could ask Leah or Barry also from Burkina.
Yeah, or someone else, please.
Did I hear Lena there? I think I heard Lena, maybe.
Hello?
Hi, guys. We do have a internet crisis here.
Go ahead, please.
Can you hear me, Frederick?
Yes. Yeah.
Okay. So in terms of this work, we have been speaking to a number of stakeholders, and really against what everyone thinks. I think this was also to our surprise. I think you can agree, Frederick, that we expected the community to be against the idea of maybe carrying and releasing modified mosquitoes, but so far we have experienced little resistance from those. They definitely need a lot of information about safety and how it can work with them, or what their role is in this technology, but there has been
We lost you, Lena. Yeah. Yeah, seems like
Do you want me to comment until she?
Yeah, go ahead, please.
Internet.
Yeah.
So there's so Lena and their team, they've published a very interesting paper. I think we've both shared it earlier in the chat about perception. They've talked to people about their perception of different technologies, and as she said, surprisingly, there was quite a positive response to the idea of modified mosquito. And if I remember well from the paper, it's related a lot to the fact you don't need to do behavior change. You don't need to like the same way for bed nets. You need to learn to sleep under a bed net. There might be also the fact people didn't understand that even with a gene drive, they would still need to sleep under a bed net because it will not address all the vectors at once, and bed nets also protect for other disease. But beyond this particular study, there's quite a lot of work in the gene drive community, some of which led by Target Malaria, but also with other teams and also researchers who are not part of developing teams of researchers. About the stakeholder engagement. So the NASEM report, the Gene Drive on the Horizon report that Fredros, you shared, mentions very clearly the need for engagement, and so does the WHO guidelines, the recent guidelines of WHO on ethics. And what these things say is you need to do engagement, you need to do better engagement that's been done so far. It needs to be more inclusive and not just about what we call a knowledge deficit model. So it's not the idea that, oh, people just don't know, but if they knew, they would love it. That's what I would say is a very old way of doing engagement. It's not very much of an engagement. This is more saying, okay, we have an idea that we think could help. What do you think? What are your questions? What do you want the researchers to answer? What are your concerns, and how can this be, for example, fed back to the risk assessment that is done? And there's been a few papers that from a wide variety of people in the sector trying to give more guidance to researchers, because the WHO and others, they're very good papers, but they're very broad. They don't give you exactly how you get to do that. So that's what we've been trying to do, and I'll put the reference in the chat.
Thanks a lot, Delphine, and thanks, Lena. In case you're about, just let us know. A quick question to Diabate, Austin, and Nolan and Tony here. When you speak to people, and especially you, Diabate, do people ask you about what will you do if something goes wrong?
Yeah, you know,
and how do you respond to this?
Yeah, I mean, this is a really frequent question that you will find, you know, across cultural different background, I mean, in Africa.
Yeah.
But to me, I mean, one thing which is really very important that we have to keep in mind that, you know, a technology should not be developed just for the sake of the technology.
Mm-hmm.
And I do believe that no one will really get excited about something that he cannot value. So if you bring in something really, and then there is a need for it, and people can see the added value of this thing to it, I think it's helped you really a lot in your engagement process. That doesn't mean necessarily that just because we know what is going to be the benefit and what is the intended use also, that systematically people are going to accept it. But at least if they see the value in it, and they feel that this is going to be important in their life, this is already halfway, you know, to acceptance. Now, beyond that, for them, you know, to formalize really, you know, this acceptance, there are a number of questions really that you need to really understand. I think that, as Delphine mentioned very clearly, it's extremely important, you know, to go to these people and then have an open dialogue. You're not just going there, you know, to convince them to say, oh, hey, it's a technology, you need to accept it. And you don't have enough information, we're going to come in to give you that information. I think it's really important, you know, to be open mind. Go to the people, have the discussion with them, let them know what you're going to do, and also have an open mind, you know, to collect all the specific concern that they might have, you know, about this technology, and then see how can you formulate this into testable research questions that you can address, and then bring back, you know, this answer to them. It's clear that if you look, for example, we have started with the sterile mail. But in many places, you know, even in some of our release site, talking to people, they will say, oh, your mosquitoes are sterile. So what will happen if you release these mosquitoes and then they biting off a wife? Are they going also to be sterile? So all these kind of questions, you might think that, oh, these are really, where are these questions coming from? But it's really very important for you, you know, to take time to understand them and then to collect them and then, you know, to address with already maybe existing literature that are out there. But if there are really some reasonable questions that can be tested and you don't have answer to this question, go back to your lab, test them. Bring, you know, this answer, you know, to your community, your public, or your stakeholders, so to make sure that you are in the same line. And as if they see that there is a really full transparency in what you are doing, and you're accountable for the work that you are doing, and you're bringing anytime that they're asking specific questions to you that are really not clear enough, that you're making all your possible, you know, to answer this question and bring them, here they are, what do you think? I think that the trust is going to be linked. There is no, I don't know how you say, a tally method, you know, to measure the level of trust. But at least if someone can allow you, you know, to get into his house for so many years of working with him, so it means that at some point there is trustability that has been built between yourself. And this is something that is critical. And we think that, you know, in the African context, because this is going to be applied in Africa, so we Africans really need to understand this, and then we can explain it in our own language to our own people. Otherwise, I mean,
on that specific point of, you know, you say we Africans, and this is an important point. In Africa today, we have the situation with the coronavirus vaccines. I know of specific cases where people say these vaccines are developed elsewhere, and so we cannot be sure that they are good. And so you see some sense of resistance based purely on the fact that the vaccines were developed somewhere else. So could you, I mean, and Tony was talking about this earlier about developing local capacity. So maybe we can start with Tony and then Diabate and Austin. Could you just describe, when you say build local capacity for management, for work on gene drive, what exactly do you mean, and how exactly do you think we can do that and now? Could you just describe this a little bit of what do you think, based on the experiences that you guys have, is the best path to building this capacity, so that when it comes to the right time, that we can actually call this thing our own?
Yeah, well, I think the capacity that you refer to, in my thinking at least, is not related only to the technical capacity to make a gene drive.
Correct.
It's the capacity to understand how gene drives work, to inform and communicate on how gene drives work, correct, to possess that knowledge, which that already exists. Actually, it's not a question of building that capacity, it's a question of leveraging that capacity of how communities are structured, what their concerns are, and how the mosquitoes behave in those communities. And there's, you know, a large amount of field entomological experience there. So perhaps on the actual, the technical GM side, there isn't as much, though there hasn't historically been as much capacity on that in Africa, and that's just a reflection of where we are with these technologies. And
do
so,
you think it's necessary for African countries or malaria endemic countries to also build capability on what you refer to as the technical side?
I think that would be welcome, yes. And you know, Mamadou Koulibaly in Mali is investing heavily in trying to build some capacity there. Diabate's lab has built capacity in how to handle these GM insects. Charles Wanji, I have, this is nothing to do with target malaria, but I have an ongoing collaboration with in terms of trying to build some functional genetics tools in Funestus. And so, yes, I think it goes to two levels. One, you know, if you can do it, you can have a better understanding of how it all works. You can either, you know, be in charge of building these types of things yourself, but you can also communicate them better and have an appreciation of the necessary data that needs to go with these type of approaches.
So you're saying, so this is important. So we need to build not just the capacity on the technical side, but also the capacity to handle and communicate and respond to this.
Yeah,
you also
it's
mentioned
hard.
Charles.
It's hard, you know, it's very hard.
Right.
You know, I'm sure there's going to be people that are watching this 2.5 hours of this thing and still struggling with some of the detail. And so it's hard to communicate that detail. It's hard, and something I struggle with is knowing, you know, when is the right amount of detail? When do you think you're being sort of patronizing by only giving the analogies and not the actual hard science? It's a very difficult balance to strike. Yeah. And so I think, you know, credit, sometimes you get credit just by showing that you're willing to do either. So there's a lot of soft skills that go with it, I think.
Thank you so much. And I mean, you mentioned Charles here, and I think this is a particularly nice thing. Charles is on the call with us. We had a wonderful masterclass with him. He's you know, set up an amazing facility there for assistance work. And at some point, you mentioned leveraging rather than building. So I guess the question here is, how do we benefit from, and maybe Diabate or Austin can answer this, how do we benefit from that existing capability to spread the know-how to handle, communicate, manage gene rev in Africa, relying on facilities that and expertise that already exist in places like Charles Wonji's lab at Ifukar in Burkina Faso, Western Kenya, Eric Uchomo's lab. You have lots of great facilities like this already.
Well, Austin, do you want to go?
Go ahead. Go ahead.
Yeah. So what I would like to say, you know, taking this a little bit back, I know that we are almost at the end of the conversation, but already as Tony mentioned it, I think it's really important to keep in mind that the gene drive technology is not just about, you know, building a perfect genetic construct. You know, because when you come out from the lab, basically you have a construct in a vial. How do you really make sure that you're going to transform that into a product to get to the field, and then when you can do the deployment? If you look along the pathway, the full entire spectrum, so you have so many professional bodies, people who are going, you know, to be involved in this. But the good thing is that if you look at the technical transformation side by itself, so in Africa, we have a really, very little capacity. Some very good laboratories are here and there. You have molecular biologists who can understand, but you have a handful of people just who can understand this. So we need really to rely on what is already there, and how do we really make sure that we can build more capacity and have a kind of critical mass of Africans who can understand this technology and who even have also the tool, you know, to do the transformation themselves in the first place. And so the liaison or the the contact, you know, with our northern partner who are mastering really this technology, they need to have a kind of transfer of technology, you know, from their side to Africa, so that we can train a critical mass of students who will have a good understanding, and then they will be able, you know, to explain it. But also, you know, if there is any specific problem, that also the solution is coming from Africa. This is the one aspect. The second aspect, you know, is we look at the full spectrum. So many other people with professional background are going to be involved. You know, be the media, for how do you communicate efficiently on this? And very importantly, also, even the regulatory landscape. If you look at the regulatory landscape in Africa, basically, I mean, you have always in many places. You have some places where they have law, but they don't have any system, you know, to regulate this. Some countries don't even have law. I think we need, you know, to cooperatively work with whoever we have to work. I think that the Nepal is doing a fantastic job there. You know, how do we really build a regulatory capacity landscape in Africa so that everybody has an understanding, and when it's come on the table, they have a say to say. They have something, you know, to say. So capacity needs also to be built at the regulatory level. Then the engagement aspect also is something which is really very critical. I used to say that, you know, you will never get in a place of someone, you know, with just your bucket of mosquitoes, just for the sake of, well, I'm a good scientist coming, you know, to release this mosquito, and then the guy will just let you release them. So the guy needs really to understand what you're doing, and then he had to be part of it. So this, but how do you really communicate effectively, you know, to these people so that there is a very good understanding. I think it's really very important. You take Burkina only, for example, we are 22 million people. How do we start even with a model? I'm bringing, a genetically modified model in Burkina. Who do I start, to engage with first? And how do I expand it? When do I involve, all these different stakeholders? They're all a specific model really has to be built there. But I think experience is already there, but it's just in the hand of few countries. How can we really take advantage of these few countries and then expand it to other countries so that people have the same knowledge and also they can approach their community the same way? And beyond that also, I think there is one other aspect, which is the operational capacity. Because we can learn all these things, if we don't have the infrastructure, to release and then follow, monitor these mosquitoes, it's useless. You will have all these things you release, but you have absolutely no way, of following them. So we need also, to build capacity in Africa, with the infrastructure at the operational level. So let's say I have a transgene. How do I handle it? How do I rare? How do I amplify? And how do I package all these things? How do we take them to the field? How am I going to release them? And how am I going to monitor all this? All these are really different aspects that capacity need to be built in. And it's going to take some time, obviously. And I believe that, the founders and all, those who are involved in this technology, the target malaria or other group, they are all aware of it. Without these different critical elements being put in place, we will never be able, to release this thing in the field.
Right. Thank you. Thank you very much. Sometimes I wonder whether this pessimism, measured pessimism, is something that you've learned after, several years, or is something that you started with in the beginning. A lot of people who hear about the technology for the first time get really excited about, something that could possibly be a game changer. But talking to you guys as experts, you are constantly emphasizing the possibility that it might not work, the possibility that it's not a silver bullet, and the fact that there's much more than just the laboratory component of it. So thank you guys very, very much. Go ahead, Tony.
I wouldn't call it pessimism. I would just call it, a healthy dose of realism. You don't want to overclaim something because it won't help your own proposed approach, and it won't help perception of that approach by others that are doing equally good work. So yeah, I would.
But if there is one,
I would call it grounded. And I think in response to your early thing, what about what if something goes wrong?
Yes.
That's a question that you can apply to anything. And so I would flip it back and say, what if we never did anything because we always worried that something could go wrong? And so we leave it with the current situation and we didn't do any interventions, then malaria would be going through the roof. So, you have a series of things. Yeah, I know. It's absolutely valid, but it's something that you apply to everything. And that's what motivates your testing of the product to try and ensure that it's safe. And then so you have an estimation of risk, and you're weighing that against the benefit, and that is what informs any decision. I think a lot of these things are exemplified because it's a new technology that has this aspect of GM and the invasiveness, and that makes people more sensitive. With some justification, but the principles don't change in my view.
Yeah, and I would just finish maybe with I would just finish with one sentence that. Somebody, said one day that, we're looking really very good. We are making very good progress. If we put our energy into our dreams rather than in our fears. We all understand, that there are skepticism really about the gene drive, but it's extremely important to work together, to make sure that we have a good understanding, and then we're addressing as we move all these specific questions along rather than, just staying there and then say, well, okay, something might go wrong maybe in 100 years from now. And then from now to 100 years, so what's going to happen? Nobody's really saying that it's going to be easy, but we have to be open-minded, and we have to be really very transparent, and also, adopt, the highest scientific code of conduct standard possible to make sure that all these different elements that are coming, whoever is doing the work, they have to be really cognizant that it's going to be accountable for what it's doing. And so it's a collective work, so we have to do it together, but we have, to take really care of, the safety issue and move side by side together and make sure that anytime be the regulator or the public are coming with specific questions and we think that these are really relevant questions, we have to go back to the lab, test them, and bring the answer.
Thank you guys very much. And I mean, this is exactly the reason I wanted to end by asking: is it a good thing that people have doubts?
Well, to me, I mean, this is the nature of anything that is new, really. I can tell you, even the cell phone.
Yes.
I know that at the beginning of the cell phone, I wouldn't use it. I was kind of a little bit skeptical. I need, to have some clear answer. If people will, oh, it's really very
now
great,
you can never get him off it.
Yeah, exactly. So you will think, well, okay, do they really understand what we are doing? I think it's really very important because this is the way.
This is a good point. And I think it's the same happening with the coronavirus vaccine in many places now. I think we can do that. Dear colleagues, I just want to thank you again very, very much. As you can see, we had lots of questions. We couldn't finish them, but this is a broad topic. I wish we had 2 masterclasses. We will talk to Diabate and Austin and Tony on how we can share some of the questions that were not answered here that you guys could help us address earlier. But I really just want to take this opportunity to say a big, big thank you for all of you. But before I do that, I would like to ask each one of you to provide any last words, last comments to the audience and to my colleagues behind the scenes who have been helping a lot and participating here. So we start with you, Tony.
It's not a concluding comment. It's just to say that I do enjoy these opportunities to engage on this, and it's nice to see that there are so many. Look down the attendee list. There's so many researchers from Africa, and I'll take this opportunity to make a plug for a PAMCA workshop that I contribute to, that's organized by Diabate and Mamadou Koulibaly and Jonathan Kayondo, which is a 3-day workshop where we talk about some of the principles of gene drives and the issues that come up with regards to that. And it's aimed at African-based researchers in this field. They don't have to be in this field, but they might be interested in it.
Thank you very much. Austin, please.
Yeah, just to likewise echo, thank you to all the attendees for your interest and the questions on this. It's always invigorating to be to discuss these things. So thanks very much for the opportunity to have this, and thank you, Fredros, for organizing.
Thank you. Diabate, please.
Yeah, well, okay. I have a last one word is that malaria is really real. So the statistic that we're having is not just number on the paper. It's really involving, a lot of suffering, and we Africans really are bearing, the cost of this suffering. And we're all cognizant that, the conventional tools that we have in hand today basically are kind of limited. Nobody's saying that we don't have to use them, but the scientific community know today that we really need new tools. And nobody's saying that it's going to be easy, but I think it has to be a collective, really, endeavor that we are all involved in. So we don't have, to set aside and then look, oh, these crazy scientists, they are developing something. Do they even know what they're talking about? They have the technical part, and then you have the other component in it. And I think we really need the collective action from everybody, to make this technology fly in Africa. And I believe that if it's fly really, it's going to make a big difference. And that will help us, to move towards the challenge of, the development that we're facing in Africa here. Thank you.
Thank you very, very much. And finally, I would like to thank all our colleagues who are on the chat box, to Charlotte specifically, and Kairos, and to Delphine. Thank you guys so much. Usually we ask any volunteering person in the audience who would like to volunteer to say a last thank you for this. So I will not wait, but if there's any volunteer, just come up right now and do that. Otherwise,
Rose, is there a way as a follow-up, because there's a number of questions that were not addressed? Yes. Do you have a way to do that?
Good question. So we have, I will send all these remaining questions to our experts We will also have a PDF version of the chat, the entire chat section, if people want to have their responses. If you need that, you just reach out to me, and I will share this with you. Does that help, Devin?
Yeah, I think so.
Okay. I think that really is it. Otherwise, for me, I'm just overwhelmed and very grateful that you guys have been able to participate. Thank you so much for your time.
Thank you very much.
Okay, great. Thank you, and bye-bye.
Thanks for organising, Fredros.
Thank you. Guys.
Thank you very much, Tony. Thank you, Diabate. Thank you, Fredros.
Welcome.
Thank you, Austin. That was really a great time. Hopefully, we're going to find out that the future of GenDrive will be bright. It's difficult, but with .NET, it starts the same way. As Tony said, with cell phone, it was the same way as well. There are a lot of questions for the unknown, but answer will come as we go along working on the field. Thank you.
Thank you.
Thank you very much. Bye-bye, everybody.
Bye. Bye. Bye, everyone.
Bye, everyone.
Bye, Fredos. Bye.
Bye-bye. I'm going to leave last, so yeah. Austin, we are grateful that we could have you for 150 minutes, maybe more. Very unusual, I imagine.
Yeah. Yeah, I think, Fredos, we can have the PDF, and if you can send it to everyone, so that we have at least a recap of the chat.
Yeah, I'll send it to you, Alex. And if anybody needs it, let me know. I can do that.
Okay. Thank you very much.